I wanted to give you a little bit of a lecture on cellular respiration, the process by which your cells take molecules like glucose and convert the energy in the covalent bonds of that sugar molecule into readily usable energy in the form of ATP within your cells. It's a complex process, and we'll try to keep it quite simple. What I've drawn here is a piece of the cytoplasm. So out here, if I can see where I'm writing, there we go. Out here is the cytoplasm. Okay. And we're starting out with the glucose molecule in the cytoplasm. In here, this is the mitochondrion. And I've drawn it very poorly, but I wanted to give myself enough space in here to draw a few things. So here we've got the outer membrane. Here we've got the inner membrane of the mitochondrion. Embedded on the inner membrane are these groups of enzymes that are involved in transporting electrons and protons in relationship to this inner membrane. And these little blue things here are supposed to be ATP synthase, the enzyme that actually makes ATP from ADP, adenosine diphosphate, and free phosphate groups. So we start out in the cytoplasm with our glucose molecule, which is a single 6-carbon molecule. And the first step in the process is to split the glucose, to split the sugar. So glycolysis is literally the lysis, the breaking of glucose. So you split that sugar in half and you end up with 2 3-carbon molecules from the 1 6-carbon molecule. And the 2 3-carbon molecules are called pyruvate. And in addition, you get 2 ATPs. And then you also get 2 alphabet soup molecules out of it, which are NAD, and I'm going to make the H on these that I'm drawing red, because we'll see that the energy we get to make ATP is associated with the hydrogen and the electrons here on these alphabet soup molecules. So that's what we get out of the initial breaking of glucose, is just allows you 2 ATPs and a couple of these alphabet soup molecules. I should put a 2 there. All right, the next step is to take the pyruvates, okay, and move them across the membrane of the mitochondrion and into the main fluid matrix of the mitochondrion and send them through a cyclical process within there where they're chemically modified, changed, and essentially all the carbon ends up being used up. So in the process of moving the 2 pyruvates across to that cycle, you lose 2 CO2s. So 2 of the 6 carbons you started with are gone as CO2. Once you get through this cycle, which is called the Krebs cycle, you lose all the other remaining 4 carbons as CO2. So once the Krebs cycle has been cranked around for this glucose, you will have converted all of the organic carbon from glucose into inorganic carbon dioxide. All 6 carbons will be gone. Now, what do we get from the process of the Krebs cycle? Well, you get some alphabet soup. You get NADHs, and you get 8 of those. And then you get 2 more alphabet soup molecules, which are FADH2, and again, energy is associated with the protons and electrons of the hydrogens on this molecule. And you get 2 of those. And you also, at some point, you get another 2 ATP. Okay. So in two major steps, we get the first step is glycolysis, which is in the cytoplasm. Glycolysis takes glucose, splits it in half into 2 3-carbon molecules. And then you take the 2 3-carbon molecules, you move them into the mitochondrion, you process those, and you eliminate all of the rest of the carbon. So you've changed glucose into carbon dioxide. It has now returned to the inorganic world from the organic world. And so far, as far as energy yield goes, readily usable energy, you've just got 2 ATP from the Krebs cycle and 2 ATP from glycolysis. That's not a lot of energy. And the mitochondrion is able to convert these alphabet soup molecules into much greater energy. So let's see how that works. What happens is NADH will donate the hydrogen and 2 electrons to this enzyme system that's embedded within the inner membrane of the mitochondrion. So 2 electrons get sent up into the enzyme system, and then they move through the enzyme system, and energy is stripped from them. And as the energy is stripped from them, that energy is used to pump protons from the inside of the mitochondrion into this space between the 2 membranes. So you get protons built up here between the 2 membranes, and then the electrons, the 2 electrons go off, and they bond up with oxygen. So you get oxygen and then these 2 electrons on it. This is why you need oxygen basically to survive. Oxygen is so ready to grab electrons that it will pull these low energy electrons from the end of this enzyme system out of there, removing them and opening the enzyme system up for more electrons to come through to pump more protons across this boundary. Now, for FADH2, what happens is FADH2 becomes FAD, and it donates 2 protons to the system, and again 2 electrons to the enzyme system. The energy from the electrons from FADH2 are a little bit lower in energy, so they have to enter the chain at a point below where the NADH electrons enter. But then they will go through the this electron transport system, and then they will also hook up with oxygen at the end. Oxygen will pull them out of the pipeline, freeing the pipeline up for more electrons. And then while that electron energy is being pumped through these enzymes, it's being used to get more protons across the membrane boundary into this inner space between the 2 membranes. So what you end up with is a bunch of protons building up on one side of this membrane. Meanwhile, you get a bunch of electrons built up on the other side of the membrane associated with oxygen. So what you've done in the mitochondrial sense, the mitochondrion has built a cellular battery. You've got a positive pole and you've got a negative pole, and now the mitochondrion is simply going to let the battery run. These enzymes here, ATP synthase, hook onto them ADP, adenosine diphosphate, that's a terrible P, and free phosphate groups, okay? And then the protons blast through this enzyme into the intermembrane matrix, or I should say just the matrix. And as they blast through, the energy of their movement literally cranks this enzyme. The enzyme, the entire shape of it cranks down and snaps ADP and free phosphate together, and that yields ATP. Then once the proton is out of the ATP synthase, the enzyme proteins rebound to their original shape. They're free then to take another proton on. The proton can blast through there again, and you can get another ATP made every time the protons are coming through here. And then once you get two protons on this side of the membrane, they can hook up with the electrons on the oxygen, and that whole system there is H2O. So the sort of waste products of generating your ATP is the carbon dioxide and the water. And remember, when we talked about respiration versus photosynthesis, photosynthesis takes these inorganic molecules, the CO2 and the H2O, and uses the energy of the sun to break their chemical bonds, rearrange the molecule, and build sugar, glucose. And then what organisms do is they then take that glucose, feed it into the mitochondrion as a eukaryote, and then the mitochondrion converts that back into ATP energy-wise, but you regenerate the original water and CO2 that photosynthesis had pulled out of the inorganic world. Now it's back in the inorganic world after you go through respiration. All right, FADH2, same kind of thing. We've generated protons. The protons pass through the ATP synthase. Two protons can then hook up with the electrons on oxygen, and again, now you've got H2O. All right, and then while the protons are blasting through, you're snapping with this enzyme, you're snapping ADP and free phosphates together, and so you're generating more ATP. So there's something that I want you to be aware of as we would take a test on this, and that would be sort of where the energy is, where the energy that we are trying to generate, the ATP, is, how it's generated. When we start out, we've got glucose, and the energy is in the molecular bonds of glucose. Our first step, glycolysis, generates 2 ATP and 2 of these alphabet soup molecules called NADH. NADH. Okay, and that's it. Then we move into the mitochondrion. Okay, so now we're in the mitochondrion. Here's the mitochondrion. And up here is the cytoplasm. In the mitochondrion, we've got the Krebs cycle, and I'm including all of this stuff from the time the pyruvates cross the membrane and get into the mitochondrion all the way through the actual Krebs cycle. For our purposes in Bio 100, just think of that as the Krebs cycle. Once you're done with the Krebs cycle, you've got 8 more alphabet soup molecules of NADH and 2 FADH2s. 8, 2, put our H's here. Okay. And then again, 2 lousy ATPs. So through the Krebs cycle, you haven't generated a lot of actual usable energy. Then you move, though, into the electron transport chain, electron transport system or chain. And when you go through that, you build the battery. So maybe what I should do is say here with an arrow, this is the battery builder. Battery builder. Then you let that battery run through ATP synthase, this enzyme that takes ADP and free phosphates and snaps them together to make ATP. And what you get out of that is 28 to 32 more ATP. So you can get a total of 32 to 36 ATP out of each glucose. And the lion's share of it is coming from the electron transport system in ATP synthase. Now, this is an extremely important reaction, and it speaks to the importance of this symbiotic relationship that must have happened long, long ago between the early eukaryotic cells and this free-living bacterium that could take the waste products from the free-living eukaryotic cell and convert them to energy. The free-living eukaryotic cell could dump its waste into the bacterium gets fed, and then the bacterium produces so much ATP there's enough to spill over and donate back to the eukaryotic cell. From the perspective of the eukaryotic cell, this is like you would crap in a box, carry that to a window, the person behind the window would weigh it and give you gold. You're taking your waste and you're turning it into the riches of energy required for metabolism. So what seems to have happened is that this relationship was so abundantly beneficial to both types of cells that eventually the bacterial cell became subsumed within the eukaryotic cell and lost its ability to live on its own, and the eukaryotic cell became so accustomed to the energy use, it was able to become more and more complex, that now if you tried to remove the mitochondria from the eukaryotic cells, they would simply not survive either. So this is how we get our energy. What I want you to know for an exam is the steps: glycolysis, Krebs, electron transport plus ATP synthase, and where the energy is in each of those steps. Glycolysis, it's in the carbon to hydrogen bonds largely of glucose and then the pyruvates. Krebs cycle, now the energy is in the electrons and protons of NADH and FADH2. You go through the electron transport system. Now the energy is in the battery that was built by the mitochondrion. The difference between the high proton concentration in the intermembrane space and the low concentration of protons in the mitochondrial matrix where the electrons still are. Then finally you let the battery run through ATP synthase, and finally you've got the energy in the chemical bond between the second phosphate and the third phosphate of ATP.